Hey guys, it's Josh with Rise Above. In a brief video, today I'm going to follow up on the results of my findings from the end of the last video. I'm currently undertaking an effort to increase my memory due to various health issues I've experienced over the past several years, which have left me with a lot of brain fog and forgetfulness. I'm taking donepezil, which is a sigma-1 agonist, and my main concern right now is currently making sure I'm not overstressing my cells. And I'll explain why right now. As previously discussed, donepezil is a sigma-1 agonist, and sigma-1 agonists are drugs or compounds which agonize the sigma-1 receptor, which is associated with various growth pathways. It tends to be anxiolytic, meaning anti-anxiety, pro-growth factor, and is known for promoting what's called cell survival. My concern here is mitigating that cell survival or prolongation of cells, which should actually self-terminate. Just making sure that I'm not inducing too much cell survival or too heavily stimulating growth pathways, etc. My way of handling this was to investigate the effects of donepezil on the mitochondria, and particularly the oxidative phosphorylation process. I read a lot of papers about mitochondria's ATP creation process, but only found 3 or 4 papers specifically regarding donepezil's effects there. Of interest, the two that were of interest were one in a mouse model of mitochondria showed a reduction of complex 1 activity. Complex 1 is the first step in the oxidative phosphorylation process, which is part 2 of ATP creation. Complex 1 is associated with the largest production of reactive oxygen species. Those reactive oxygen species are basically uncoupled electrons which are responsible for oxidative stress. I explained in the last video that these are normal part of functioning physiology and are expected in some capacity, but when you have too much of it, that's when you get oxidative stress essentially. So that was the first study. The second one was a model in pig brains showing an increase in complex 1 activity. So the first study showed a decrease, and the second one showed an increase. And in that pig model, there was an increase of complex 1 activity and reactive oxygen species, but overall a reduction in amyloid beta generated reactive oxygen species. That second one was actually meant to simulate an Alzheimer's symptomatology, because amyloid beta is the primary protein which builds up in the form of plaque in the brain, contributing to Alzheimer's, and is also considered a hallmark symptom of Alzheimer's. The other 2 studies I mentioned, one was about the effects of donepezil on skeletal tissue, and the other was about neuroprotective properties of sigma-1 agonists. So based on these, I'd say inconclusive results, the jury is still out on this. Ideally what you would want is human trials, either in vitro, which is, under test conditions of certain tissues or cell lines, maybe in tubes or, what have you, in some kind of culture, or in vivo, which is actually inside the human body. Since those aren't available under this very specific research that I was looking for, I can extrapolate from these animal models and kind of make an educated assumption, right? Not something you want to do with certain types of compounds, but given melatonin is well known to be safe, I think I can actually move forward and add that as an antioxidant into my stack. Given that reactive oxygen species tend to be elevated in unhealthful conditions, I think that's probably a good way to go. I've compiled a lot of studies on melatonin, which according to the literature has neuroprotective, cardioprotective, and possibly some cancer preventative properties. Of course, as is the thing with many treatments or potential treatments for cancer, obviously nothing is clear. I've compiled many studies on melatonin, which according to the literature has neuroprotective, cardioprotective, and possibly some cancer preventing properties. Some possible cancer preventing properties. While the literature on melatonin is huge, there's nearly 30,000 papers on the subject, I will present the findings on the effects of melatonin as an antioxidant, as well as its neuroprotective, cardioprotective, and anti-cancer qualities in my next video. We'll also cover recent news articles that were brought to my attention that have come out in the media about concern over melatonin's increase in use for long-term sleep assistance, which rose apparently from 0.4% to 2.1% of melatonin users in the last 20 years. Whether that concern expressed in these articles is warranted, I'll be researching any longitudinal studies available to try and answer that. So just a quick one today, just checking in, and I hope you guys are well, and I'll catch you on the next one.